Hemos becado a más de 1 millón de estudiantes universitarios.
Más de 84,000 alumnos y alumnas han sido beneficiados con becas nutricionales.
Lanzamos una campaña para apoyar a la Universidad Autónoma de Guerrero, afectada por el huracán Otis.
Este año organizamos nuestra tercera gala musical y nuestra cuarta carrera atlética.
Estamos felices porque más del 87% de nuestros becarios son la primera generación de universitarios en su familia.
Cada año se apoyan a 100 estudiantes de la Facultad de Derecho.
Hemos entregado más de 600 becas a estudiantes de bachillerato junto con Fundación BBVA.
Hemos transmitido 195 documentales y 160 conferencias.
Apoyamos a la Unidad de Investigación en Neurodesarrollo de la UNAM.
Todos nuestros informes son públicos.
Se han entregado más de 50 premios para fomentar la investigación científica.
Con el programa Bécalos, el Patronato de la Facultad de Química y Fundación UNAM otorgaron más de 30 becas.
Con la ayuda de Fundación Carlos Slim hemos otorgado becas de apoyo nutricional y económicas.
Con el programa Hídrico Modelo ayudamos a 340,000 habitantes.
Tenemos 2 premios EFI Social por nuestras campañas publicitarias.
Junto a Grupo VAL, más de 2,000 universitarias han sido beneficiarias.
Otorgamos más de 400,000 tratamientos.
Durante la pandemia apoyamos con las campañas Dona un Kit y Apoya a un Residente a estudiantes de medicina de la UNAM.
Durante el confinamiento COVID, donamos tablets para estudiantes de escasos recursos.
Hoy son más de 27,000 solidarios impulsando la educación.
Participamos en la conservación de corales en el Caribe mexicano.
Brindamos más de 4,000 becas de movilidad internacional.
Colaboramos con el Semillatón a preservar el maíz en la Sierra Tarahumara.
Apoyamos el proyecto de conservación de perritos llaneros en la Reserva de la Biosfera de Jano, Chihuahua.
Participamos en el proyecto de conservación de la guacamaya roja en el Parque Nacional de Palenque.
Desarrollamos el programa Líderes Transformadores.
Participamos con el Consorcio de Universidades por la Ciencia.
Organizamos 11 ediciones del Foro 2020.
Fundación UNAM resguarda el Palacio de la Autonomía, inmueble histórico de la universidad.
A través del CI ofrecemos cursos, diplomados y talleres.
Dotamos de equipo al Observatorio Astronómico Nacional de San Pedro Martes.
Apoyamos a alumnos de escasos recursos.
Asignado a la opción señalada, institución de asignación UNAM.
¡Sí quedé!
El sueño se cumplió. Ahora formas parte de la comunidad universitaria y puedes acceder a las becas que Fundación UNAM tiene para ti. Conoce nuestros programas en www.funam.mx y en nuestras redes sociales.
asignado en la opción señalada, institución de asignación UNAM.
¡Sí quedé! ¡Sí quedé!
El sueño se cumplió. Ahora formas parte de la comunidad universitaria y puedes acceder a las becas que Fundación UNAM tiene para ti. Conoce nuestros programas en www.funam.mx y en nuestras redes sociales.
Asignado en la opción señalada, institución de asignación UNAM.
¡Sí quedé! ¡Sí quedé!
El sueño se cumplió. Ahora formas parte de la comunidad universitaria y puedes acceder a las becas que Fundación UNAM tiene para ti. Conoce nuestros programas en www.funam.mx y en nuestras redes sociales.
Bienvenidos de conferencias de investigación universidad organizado por el Colegio Nacional y el Consorcio. El día de hoy tenemos Ciencia, nos años Welcome to organized by Fundación and the Today we have with a professor. Welcome. Is lecture. The conference is the part cycle Genomics. They are characterized by a rapid advances about the cycle UNAM English. Moorjani has been studying the evolution of the humans for a long time with very outstanding results. We are extremely happy to have Priya with us today. Nos acompañan en el panel el presidente de Fundación UNAM, Dionisio Meade, la presidenta del Consorcio Universidades por la Ciencia, Araxi, Urrutia, y nuestra invitada especial, la doctora Priya Moorjani. We are joined by the president of the UNAM Foundation and the president of the Consortium of Universities for Science, organized a few years ago with universities and research centers in Mexico, the US, UK, Canada, Spain, Brazil, and it has been extremely successful. This seminar cycle has been part of the activities of the International Year of Basic Sciences, organized by the United Nations and the UNESCO, and now it's the Decade of Sciences for Sustainable Development. We welcome you, and as always, we invite you to make questions, comments through the platforms of the Consortium and the UNAM Foundation. Pasamos ahora la palabra a nuestro presidente de Fundación UNAM, Dionisio Meade, y luego Araxi nos hará la presentación de la doctora Priya Moorjani.
Muchas gracias, Jaime. Thank you very much, Priya, to be with us, and we are very interested. We thank you very much for your cooperation with this consortium. Esta pues es una prueba más del éxito que tiene este consorcio, de una gente egresada de Harvard, ahora está en la Universidad de California, suma a nuestras presentaciones. La verdad es que nos sentimos muy motivados. Ya son muchas las series cada semana damos algún documento, una presentación, alguna investigación siempre. Algunos de quienes acompañan del consorcio. Siempre nos da mucho gusto que nos acompañen, pues algún egresado de la UNAM, experto académico, nos da mucho gusto. Pero cuando es, como el caso de Priya hoy, de fuera, ¿qué nos motiva especialmente? Quiero mencionar que formamos parte de un comité asesor de Naciones Unidas a las ciencias básicas. Y precisamente las Naciones Unidas nos invitaron, la UNESCO, todo por considerar que programas como estos eran programas que eran muy convenientes, muy favorables a divulgar la investigación científica. Siempre nos motiva a pensar unos de nuestros clientes, después de estas presentaciones, como ahora en esta serie biogenómica, pues puedan interesarse por los temas que aquí se tratan. Precisamente el sentido de estas presentaciones, ese espacio de difusión, que se suman pues muchos oyentes, tanto de la propia universidad, los centros universitarios que tenemos fuera. En este caso estamos seguros que también alumnos y estudiantes de la Universidad de California están acompañados. Así que pues por todas estas razones nos motiva mucho esta discusión. Agradecemos a la universidad por su apoyo, agradecemos al Colegio Nacional en el evento, agradecemos desde luego a Jaime a favor de dar este proyecto. Hoy nos va a estar con nosotros, pero está Priya y agradecemos mucho, Priya, que nos acompañes la tarde de hoy. Muchas gracias a todos, buenas tardes y pues con el mayor interés de escuchar tu presentación. Gracias, Priya, y gracias a todos los demás, gracias a quienes nos dan apoyo técnico para realizar esta presentación y que podamos avanzar con esto. Muchísimas gracias a todos.
Muchas gracias, Dionisio, y también al doctor Jaime Urrutia, Dionisio, presidente de Fundación UNAM, por sus palabras. Y bueno, pues estamos vestidos de gala otra vez.
Very much for the invitation. This is such an exciting series. It's really my pleasure to be able to contribute to this series and share my lab's research with. I'll start by telling you a little bit about my research. My lab's research focuses on studying human evolution. We are interested in trying to understand where humans come, how we are related to different populations, individuals over time, and what are some evolution events, founder events or selection that have occurred during our when they have occurred, and which mutations drive human adaptation disease. To answer some, we use DNA. DNA, you know and have heard from previous speakers, is present in all the cells in our body, but it is really unique because it inherited from our ancestors. Of us has half of our DNA, parents inherited half of their from their parents, who inherited half of their DNA from their parents. And so if you take DNA from any of DNA of one individual, and then paint their genome, segments of DNA coming from different ancestors, what we see is a beautiful mosaic. Here I'm showing DNA of an individual, and I'm showing you their 20 chromosomes as well as their sex chromosomes, X chromosomes. And I've colored the genome based inherited from different ancestors. And you can see a beautiful mosaic with different parts of the DNA coming from different ancestors. We can also look at specific regions or mutations that are present in these in the DNA to then understand the function of those regions. For instance, we know that a region on chromosome 7 may have contributed to language acquisition in humans, and we can also study other regions across the genome, which can also tell us about the risk of getting different diseases. And this is the kind of information that we use in our lab to understand ancestral population relationships, as well as to understand different variants related to human adaptation and disease. Now, this is, as you've heard from many other speakers, the most exciting time to do human genetics because there has been a major explosion of data in the field. We now have hundreds of genomes from diverse worldwide populations and thousands of genomes of different individuals, including whole genome sequences from many different groups. There is more than 6,500 genomes in the Mexican Biobank study. And one of the largest study among these is the NIH's All of Us study, which aims to collect 1 million genomes of people living all over America. And this kind of data is really allowing us to more precisely investigate how different people are related to each other and also to understand disease susceptibility. But what is also really exciting in our field is that now we have figured out a way to look in the past. And this approach is the study of ancient DNA, which has given us a time machine to try to see directly in the past. This idea of studying ancient genomes was pioneered at UC Berkeley, and so I feel incredibly fortunate to be doing my research at University of California today. Alan Wilson, who was a pioneer in human genetics, had the insightful idea of using DNA from ancient samples, and he sequenced the first quagga genome, which is an extinct species, and tried to understand why the species may have gone extinct and how it relates to present-day living species. Svante Pääbo, who did his postdoctoral research at University of California, then expanded this analysis to looking at ancient DNA samples, looking at ancient DNA of human samples by first sequencing the Neanderthal genome, and through his work also discovered a new hominin species, the Denisovans. And today this field of ancient DNA has more than 10,000 genomes that have been sequenced, and this data is really changing and transforming our understanding of human history and evolution. You heard about some of this work also from the other speakers like Vagish Narasimhan and Mansa Raghavan, who also talked about how this is really helping us understand how different people related to each other in the past, and also to study human adaptation over the past. And this kind of data is really transforming our understanding of human history, and it's also providing an atlas of genetic variants that help us understand human health and disease, and also how evolutionary history has impacted human health and disease. And so today in my talk, I'll tell you about three stories. In the first story, I'll tell you about gene flow between modern humans and Neanderthals. In the second story, I'll briefly touch upon how the events of the Neolithic have transformed our genomes and have shaped genetic variants in our genomes. And then in the third story, I'll tell you about how, using whole genome sequences from India, we were able to reconstruct the evolutionary history of South Asians and study adaptation and disease on the subcontinent. So the first story is looking at Neanderthal ancestry through time. In this case, this work was led by a postdoc in my group, Manjusha Chintalapati, in collaboration with Leonardo Ayasi and Ben Peter at Max Planck Institute. Now, just to provide you with a little background, as you saw from Professor Brenahan's talk and from Sarah Tishkov's talk, modern humans in Africa. We know this because the largest diversity. And then about 50 years ago, a group of humans left Africa and populated the rest of the world. We know modern humans got to Europe around years ago, also South Asia, and about 15,000 ago, almost parts of the world were inhabited with the settlement of the Americas also. Now, in this journey, modern humans were not alone. Through the sequencing of the Neanderthal genome, we now know that modern humans met with Neanderthals when they left Africa, and as a consequence, today, all non-Africans have about 1 to 3% Neanderthal ancestry. And as I mentioned, through the sequencing of the Denisovan genome, actually the sequencing of a tiny little pinky bone that was discovered in the Denisova cave, we now know that people living in Oceania and Asia also have some ancestry from this Denisovan group and have inherited about 0.1 to 5% ancestry from this group. Now, for this talk today, I'm mainly going to focus on Neanderthal ancestry for this project. What has been really puzzling is that we know that all non-Africans have Neanderthal ancestry because we believe that the gene flow occurred in the common ancestors of modern humans as they left Africa. But if we look at Neanderthal ancestry variation across living individuals, what we find is that people who live in Europe today or South Asia or the Americas tend to have about 1.5% Neanderthal ancestry. But if you look at people in East Asia or Oceania, they tend to have about 20% higher Neanderthal ancestry, around 2% of Neanderthal ancestry. We can also look at the variation in Neanderthal ancestry across the genome by looking at different regions of the genome and estimating how much Neanderthal ancestry is present in these regions. On average, most regions have about 1 to 2% Neanderthal ancestry. Here, I'm showing you a cartoon of a region across the genome where we can look at each individual and mark where they have Neanderthal ancestry in blue. On average, you can see each of these individuals would have each of these regions would have about 1 to 2% Neanderthal ancestry. But in some regions of the genome, many Neanderthals who had been living outside of Africa for about 300,000 years before modern humans came, and they were adapted to the extreme climates of Europe during the Ice Age. So they were adapted to the climate as well as the pathogens, and so it's possible that modern humans inherited some variants through Neanderthal gene flow that allowed them to adapt to the pathogens outside Africa. Interestingly, also, there are regions of the genome that are completely devoid of Neanderthal ancestry. There are at least 5 to 6 regions in our genome that are very large, greater than 10 megabases long, that are lacking any Neanderthal ancestry. And these regions are often referred to as deserts of Neanderthal ancestry. So this leaves us with some open questions of, was Neanderthal ancestry a single event in the common ancestors of all modern humans, or did it occur multiple times? And if it did, then what was the timing of these different events? Also, is Neanderthal ancestry neutrally evolving across our genome, or has been adaptive or deleterious for modern humans? And so to answer this question, we use this key insight of using DNA clocks for trying to learn about the timing of different events. Now, as you know, when diverse groups like, say, Neanderthals or modern humans would mix with each other, the next generation would get one chromosome from each of the parents. So you get one chromosome of Neanderthal ancestry, one chromosome of the modern human ancestry. But what happens during meiosis is that the DNA of the parents gets mixed up because there are crossovers that occur in 1 to 2 positions in each chromosome, such that now there is mixing of the blue and the gray ancestry, and because of these crossovers. And these crossovers continue over time, and in each generation you get 1 to 2 crossovers per chromosome, and then these blocks become smaller and smaller as a result, and now you have little blocks of blue ancestry in the gray background and little blocks of gray ancestry in the blue background. So the key insight is that the lengths of these blocks can work as a clock for trying to date different events. So if we take a genome and we color it based on Neanderthal ancestry, and if we find very short blocks of Neanderthal ancestry, then we know the mixture must have occurred a long time ago. And if we find very long blocks of Neanderthal ancestry, then we know that the mixture must have occurred more recently. But given that now we have ancient DNA samples, we can do even better, and we can not just look at present-day human samples, but also look at ancient DNA samples. And what is really cool with ancient DNA samples is that these are samples that might be closer to the Neanderthal gene flow event. And so we might be sampling over here just few generations after the gene flow. And when we look at present-day individuals, we are sampling over here many generations after the gene flow. So we expect there to be small blocks of Neanderthal ancestry in their genomes. And this is the key insight that we used to try to learn about the dynamics of the human-Neanderthal mixture. In our study, we had data from early modern human samples. We had data from 59 ancient DNA samples that were dated that were collected that were dated over the past 45,000 years. We have some samples that are 45,000 years old, other samples that are 10,000 years old, 20,000 years old, 2,000 years old, and as well as we have 300 samples of present-day individuals that have been sampled all over the world. We also then take data from 3 Neanderthals as reference samples, as well as from 200 sub-Saharan African individuals who have negligible amounts of Neanderthal ancestry. And what our goal in this study was, one, to try to date when the Neanderthal mixture occurred. And to do this, we do chromosome painting. So we take these ancient and modern samples and we compare the DNA with sub-Saharan Africans as well as Neanderthals and try to paint the genome at every position in the genome. Here the Neanderthal ancestry is being shown in blue, and we can actually identify at every position in the genome what is the probability that the individual has Neanderthal ancestry or they have modern human ancestry. And then we can do this analysis for both ancient and present-day samples, and then look at the distribution of those blocks to try to infer when the mixture occurred. So if you find very large blocks of Neanderthal ancestry, you know that the mixture must have occurred recently. And if you have very small blocks, then you know that the mixture occurred a long time ago. Another thing we can do is also look at how Neanderthal ancestry has changed over time. Because we have samples collected all over time, we can see between, say, 50 to 40,000 years, what was the frequency of Neanderthal ancestry compared to the frequency between 40 to 30,000 years ago, or the frequency at present, and try to look at the trajectory of how the frequency of the variants has changed over time. And this can also allow us to understand if Neanderthal ancestry was under positive selection or negative selection, because a variant that is very rapidly increasing in frequency might be indicative of something might be indicative of a beneficial variant that is increasing because of selection rapidly in the population. So let's look at what our data looks like. Here is a sample called Malta. This is a 24,000-year-old ancient DNA sample that was found in Siberia, what we first did is an earlier study had sequenced the Malta genome to about 1 to 2x coverage. We took this data and then we tried to color the genome of the Malta individual. Here are the 22 autosomes of Malta and the X chromosome of Malta. And then we color each of the positions with the probability of having Neanderthal ancestry. So the blue blocks are telling us where this individual harbors Neanderthal ancestry. Based on these little blocks of Neanderthal ancestry in the Malta genome, we can infer that the gene flow, the Neanderthal mixture in Malta occurred about 26,600 years before the individual lived. Note that when an ancient DNA individual dies, these genetic clocks stop when the individual dies. And so if you want to estimate the time before present, you have to add the age of the individual back. And by doing that, we infer that the Malta gene flow occurred about 47,500 to 46,700 years before present. We can apply the same analysis to another sample. This sample is Oasa. Oasa is a 42,000-year-old ancient DNA sample found in Romania. This is a really unique sample because Oasa is an interesting sample that has morphological traits that are intermediate between modern humans and Neanderthals. And so when this individual was sequenced and we looked at Neanderthal ancestry in the Oasa genome, what we were surprised to see is that this individual has very large blocks of Neanderthal ancestry in their genome. And this is because the gene flow in Oasa occurred about 200 years before the individual lived, which means that the time before present would be 44,700 to 37,000 years before present. But 200 years before 200 years is actually only 6 generations. And this means that Oasa is a great-great-grandchild of a human-Neanderthal mixture. And its samples like Oasa sort of clearly show us that there must have been gene flow between modern humans and Neanderthals. We can actually apply this kind of analysis to many of the other ancient DNA samples that I mentioned. Here I'm showing you the estimate of the different samples. On the x-axis is the sampling age, often based on radiocarbon dating for the sample. For some samples, you can see that the radiocarbon date is very wide and uncertain. On the y-axis, I'm showing you the time of the gene flow that we infer based on using the DNA clocks. And we find that there is a clear relationship between the two, and we can fit a linear model, and we infer that the mean time of the Neanderthal gene flow across all these samples is about 47,000 years before present. Now, this model assumes that there was only one interaction between Neanderthals and modern humans. What if there was multiple pulses of mixture between Neanderthals and modern humans, or what if the gene flow happened over a long period of time? So to answer this question, what we did is we tried to integrate the information across all the samples and tried to see if a model with a single or multiple pulses or a long duration is a better fit to the data. What we find is that a model with a single pulse is a better fit to the data, but actually not a short duration, but a long period of gene flow between the two groups. So a duration of gene flow that was almost 7,000 years long. And what almost 7,000 years long. And this means that the most likely period of Neanderthal gene flow is between 43,500 to 50,500 years ago. The next thing we wanted to also do is to try to see when these deserts and peaks were formed by looking at Neanderthal ancestry in different time scales across our samples. And so what we did is we looked at the frequency of Neanderthal ancestry across the genome. Here I'm showing you a region from chromosome 9. On the top part of this plot is the frequency of Neanderthal ancestry across our ancient DNA samples in blue. And on the bottom is the frequency of Neanderthal ancestry in the present-day individuals in our data set. We can then look at regions like this where Neanderthal ancestry is present at high frequency, both in present day as well as ancient DNA samples. Or we can look at regions where it's present at high frequency in ancient DNA samples, but not present-day samples, or the other way around, ancient samples and not present-day samples. Let's look at one of these regions. This region is BNC2. It's present at high frequency both in our ancient and present-day samples. We can then look at the trajectory of this variant by looking at the frequency in different age bins. So if we look at only our samples that are dated between 45 to 30,000 years, we find that this variant is present at about 15% frequency. We can look at also 10,000 to 1,000 years, and now it's present at a higher frequency. And if we look at modern human samples, it's present at nearly 60% frequency. Now, BNC2 plays a role in keratin biology, and from this trajectory, we can see that it was immediately selected after the gene flow. We can also look at other regions where some regions we find that the frequency is not very high early on, but in our most recent samples, the frequency is much higher. And this might be suggesting that as the environments changed, this variant that was initially present was not particularly beneficial, may have become beneficial and rapidly increased in frequency. And consequently, conversely, we can also see that some variants that may have been present at very high frequency early on did not seem, they might have carried some deleterious variants, or they might be linked to other deleterious variants, and over time their frequency may have decreased in present-day individuals. We can also look at how these Neanderthal ancestry deserts were formed. And so to do this, what we did is we looked at Neanderthal ancestry across our time stratified samples, looking at samples from 45,000 years to present day. And we looked at some of the regions of deserts that had been previously identified. As I mentioned earlier, there are 5 regions that are greater than 10 megabases where in present day individuals you find no Neanderthal ancestry. Across different studies, the boundaries of these deserts have been slightly different, including in our study. And so I'm showing you the region between these dark lines, which is the boundaries from our study or from other studies. And then we can see if there is any Neanderthal ancestry. If we confidently call Neanderthal ancestry, then that's shown in dark blue color. And if we find short blocks of Neanderthal ancestry, that could be just because of ancestral polymorphisms shared between modern humans and Neanderthals. Those are shown in light blue boxes, and the gray regions mean just modern human ancestry. And as you can see for this desert on chromosome 7, there is no Neanderthal ancestry even in our very earliest samples that are dated to 45,000 years. What this means is that Neanderthal ancestry in this region may have been so deleterious that it was not tolerated and was immediately purged out. Conversely, it could also mean that modern human ancestry in this region was so beneficial that it was beneficial to retain modern human ancestry in the region rather than to harbor Neanderthal ancestry in the region. We can also look at other deserts. There's another one on chromosome 8, 13, 3, and 9. And in most cases, you find very little Neanderthal ancestry, if any, in any of the individuals. On chromosome 1, you start finding some individuals have some blocks. On chromosome 3, we find that there is Neanderthal ancestry, but just outside the boundaries of the deserts that we identify. And this suggests that these Neanderthal ancestry deserts were formed immediately after the gene flow, as has been predicted theoretically also, but now empirically we can also see in the data. Now, so to just summarize our findings from this study, I showed you how all non-Africans have Neanderthal ancestry. And in this study, we were able to infer that this might be because of gene flow that occurred between 43,500 to 50,500 years ago. We find that there was selection, both positive and negative, that occurred rapidly to form these deserts, as well as in many cases rapidly to increase the frequency of some beneficial variants acquired through Neanderthal gene flow. Now, when we published we were really excited to find actually at a conference a talk that was given by our colleagues at Max Planck Institute, where they had sequenced 2 new ancient DNA samples. One sample's Laticune, which is 45,000 years ago, and another sample from Ranus, which is a 25x coverage genome that was dated to 42 to 49,000 years before present. By using these similar techniques for genomic dating, they also inferred that the Neanderthal gene flow occurred between 45,000 to 49,000 years before present, with a mean of 47,000 years, as we had inferred in our study. So it was really exciting to see sort of replication from another group with some new samples and to converge on similar estimates. But one day, like I was also looking at YouTube, listening to talks related to Neanderthal ancestry given by a world expert, Tom Heim from Vienna, where he was discussing some of their studies of looking at dates of Neanderthal samples that had been collected in different regions in Eurasia. Now, here they had done dating, which was radiocarbon dating rather than genomic dating. And using that information, they were trying to estimate the spread and the presence of Neanderthals in different regions across the world. And just to show you some example of this, if we look at some of the samples from, say, Northwest Spain, they inferred that these samples were dated, accounting for the uncertainty, between 50,000 years to about 40,000 years before present. Using this kind of information, they had dated that the last Neanderthals lived in Eurasia about 41,000 to 39,000 years before present. But what they have, their group, including others like Katrina Duca and Jean-Jacques Hablan in France, have started doing is that they are also now looking at the overlap of Neanderthals and modern humans in the same regions, and have now, using that information, inferred that there was potentially a 6,000 to 7,000 year long overlap between modern humans and Neanderthals in Eurasia. And this is really interesting because even based on our genetic dating, we infer that modern humans and Neanderthals must have been exchanging genes for about 7,000 years. And then from archaeological data, slightly looking at almost independent information, they are dating that the overlap of the two groups was also around 6,000 to 7,000 years. And what this tells us is that the separation of these groups, or the disappearance of Neanderthals, may have been far more complicated and than we had previously appreciated, where modern humans and Neanderthals were not so different that they would have just outcompeted each other, but in fact they exchanged genes and were living together for a long period of time, and slowly maybe they gradually got assimilated in our population. With that, I'll switch to the second story. In this case, I'll tell you about how we can use these similar ideas for learning about the timing of events at more recent time scales, in particular during the Neolithic farming. So if we look at the availability of ancient DNA samples across the world, what we see is that by far majority of the samples are actually from more recent time scales. We find that majority of the samples date back to about 10,000 years. And in fact, there are very few samples that are older than 20,000 years. This is why in our previous study, we only had 59 samples that are between 20 to 50,000 years, whereas majority of the samples, and in fact about over 10,000 samples, have now been collected for ancient DNA, and majority of these have been collected in for samples dating to the past 10,000 years. And these samples have been extremely useful in trying to expand our understanding of the impact of Neolithic farming and the recent events in human history. And so we wanted to expand our dating method that I showed you earlier for dating Neanderthal mixture in trying to date events at more recent time scales. So we developed this method, dates, and try to apply it to samples from Europe. So in this case, as you've seen from other speakers, if we take DNA from different regions in Europe, what we see is that Europeans actually have a three-way mixture. Europeans have hunter-gatherer ancestry from groups that were indigenous to Europe, lived there about 10,000 years ago. This is shown in blue. Europeans also have ancestry from Anatolian or Near Eastern farmers. This is because of a migration from Anatolia about 7,000 BC ago. And most Europeans have this orange ancestry deriving from Neolithic farmers. And more recently, people also have ancestry from steppe pastoralists. Now, as many of the speakers mentioned, the sampling in Europe is now very fine scale, where you can get multiple, where it's typical to collect time transects of data from similar regions. So here is an example of one of these time transects. And in this case, each of these dots is a different sample from Hungary. There are more than 17 sites where samples have been collected in Hungary, and these samples date between 5000 BC to 2000 BC ago. And so, and if you look at the ancestry of these individuals, what you find is most of these individuals carry some amount of Anatolian farmer ancestry, as well as the hunter-gatherer ancestry that is indigenous to this region. So we applied our genomic dating method to then try to infer the timing of the Anatolian gene flow in these groups by applying it to each sample separately. And what you see is archaeologically, the evidence for farming dates back to about 7,000 BC, which is shown here in the dotted line. Here on the x-axis are the radiocarbon dates. On the y-axis are the times of gene flow that we infer based on dates. And then we can apply this method to each sample separately and infer the timing of the mixture. And what is really striking is that we find that the first earliest timing of gene flow is about 6,500 years ago. This means that farming arrived 7,000 BC ago, but for about 5,000 years, there is no mixture between these groups. And then all the groups present afterwards appear to have both the Anatolian and the hunter-gatherer ancestry. What is even more interesting is that if you look at the dates in the younger samples collected about 2000 of 2000 BC age versus our older samples, the 6500 BC age, what we find is that in the second case, we infer much more recent dates compared to the earlier samples. If we look at the ancestry profiles of these individuals, we also find that there is higher hunter-gatherer ancestry in the more recent samples, which is also interesting because this is the timing is more recent, because what we are seeing is that there was a resurgence of hunter-gatherer ancestry. So after the initial mixture, certain groups got isolated and then had secondary pulses of hunter-gatherer ancestry. And that's what we are seeing in the more recent samples. And this is how we can use ancient DNA, not just to learn about the timing, but also the dynamics of mixture between different groups. Now, this is not an isolated event in Hungary. There is now data from many different regions across Europe, and one can apply the same kind of analysis for different regions, including from Northern Europe or Southern Europe, and then try to see the dates of the different admixture events. And what we infer is that the gene flow between hunter-gatherers and farmers occurred between 7,000 to 4,000 BC ago for a long period of time of mixing of nearly 3,000 years. But what is interesting is if you look at present-day individuals, they actually have a third ancestry, and this is the steppe pastoralist ancestry. This ancestry is not seen in our ancient DNA samples from 4,000 or 5,000 years, 4,000 or 5,000 years ago. But then when you start sampling more recent individuals that are sampled 2,000 years ago or 3,000 years ago, you find that they do appear to have this steppe ancestry. This is because the steppe gene flow occurred between 3,500 to 2,500 years ago, and it rapidly spread across Europe. We can look at samples from the late Neolithic or Chalcolithic or Bronze Age, and now all of these samples have the steppe ancestry. And today, in many groups, the steppe ancestry is actually the majority ancestry that was not present even 5,000 years before present. And this means that these technological developments like Neolithic farming or steppe ancestry, which is associated to these herders who lived in Russian steppes and Ukraine, and may have been responsible for domesticating horses and potentially spread Indo-European languages across the world, have had a massive impact on the genetics of the people living there also. But this is not an isolated event. If I uncover the other part of this map, you will see that South Asians too have the same two mixtures. They too have Neolithic ancestry. This is more related to Iranian farmers than Anatolian farmers, as well as steppe ancestry. And again, we can use genomic dating methods and try to look at ancient DNA samples from South Asia. And here we date that these mixtures occurred more recently in South Asia than in Europe. So Neolithic farming comes about 5,400 to 3,700 BC, and we date that the steppe gene flow occurs about 1900 to 1500 BC ago. So these events are more recent in South Asia, but are still the same ancestries. And this shows that how people living across the world, while they may be distinct, actually are also much more similar than we think. And also, historically, many of the same events have impacted all of us living around the world. And now finally, I'll tell you a third story where we are looking at genomes of present-day Indians. And in this case, I'll try to focus on how we are reconstructing evolutionary history, but also how that informs us about disease and adaptation. Now, in this study that was led by Elise Kurdenkopf and Lauritz Koff, who are two postdocs in my group, in collaboration with Dr. Sharon Cardia at University of Michigan, Professor Jin Cookley at University of Southern California, and Dr. A.B. Day at the All India Institute of Medical Sciences in New Delhi. We collected data for 2,700 samples, as well as extensive phenotype data. All the collection was done by Dr. Day at the All India Institute of Medical Sciences in India, with a fantastic field team that collected data from all over the country in a very careful and thoughtful way. And we then sequenced these samples to generate whole genome sequences for 2,700 individuals from different regions across India, people speaking different languages or practicing different cultural practices. And we then took this data and tried to see how this three-way model that I just told you about, how does that fit the data of people living in different regions in India, if they were sampled from north, central, west, east, or south. And what you can see is all, regardless of the region individuals come from, they have the same three-way mixture. What is really interesting is that there is some variation in the proportion of the mixtures. So people who live in the north appear to have higher amounts of the steppe pastoralis ancestry compared to people who live in the south. People who live in the south appear to have higher amounts of the South Asian hunter-gatherer ancestry compared to people who live in the north. Now, one consequence of the steppe ancestry in Europeans has been lactase persistence. Lactase persistence, which you heard about from the talk by Professor Vagish Narasimhan, is the ability to digest milk in adulthood. Now, most of us are born with the ability to digest milk, but once we are 4 or 5 years old, we lose this ability after weaning, like most mammals. However, some individuals have a mutation upstream of the lactase gene, which keeps this gene constitutively on in adulthood, such that they continue producing lactase and can digest milk even in adulthood. Now, this mutation is the strongest signal of selection in our genome, and that's because in some population, this mutation is present at a nearly 90% frequency. So if you look at Northern Europeans, this mutation is present at nearly 90% frequency. It's at slightly lower frequency in Southern Europeans. African populations have other mutations in the upstream of the lactase gene that allows them to digest milk in adulthood. Now, given that and from ancient DNA analysis, now we know that this mutation actually arose in these steppe pastoralist groups. And so we wondered that given that South Asians have a similar ancestry profile as Europeans, do they indeed have these variant present? Do they indeed have the lactase persistent variant also? And so if we look at this variant, it is indeed present in India, and it is present between 0 to 30% frequency. It's present at a higher frequency in northern India than in southern India. But what is really striking is that in two pastoralist groups in India, one from the north of India, Gujar, and one from the south of India, Toda, it's present at nearly 90% frequency. And when we looked at the region around the lactase gene in these pastoralist groups, what we were really surprised to see is that actually there is a nearly identical haplotype that is present in the Toda and Gujar individuals, same as the steppe pastoralist ancient DNA samples or the Great Britain individuals from UK. This means that the same haplotype is present in both of these groups, and both of these groups have inherited this lactase persistent mutation from the steppe pastoralist groups, highlighting how they've adapted to different environments with the same mutation. Now, in the past 5,000 years, there has been a demographic shift in India where groups that were previously mixing with each other, like related to these South Asian hunter-gatherers and Iranian farmers and steppe pastoralists, are now tend to marry more endogenously, and so we find that different groups are much more distinct to each other compared to what they might have been in the past. Now, one other way we can look at this is we can look at each individual and we can look at what proportion of their genome is present in runs of homozygosity. Now, as you know, if two parents are related to each other, then the probability that you inherit the same piece of DNA from both of them is much higher. And so then the DNA in the offspring at that positions would be homozygous. We can look at what proportion of the genome is homozygous across different regions in India, as well as in worldwide populations. When we look at Africans, for instance, we find very little homozygosity across their genome, which, as you might remember from Professor Branahan's talk, that there is large diversity in African populations. Similarly, we see much less homozygosity in Europeans and East Asians. However, in some regions, individuals, especially living in the South, have high rates of homozygosity in their genome. And this can be because of consanguineous marriages, which might be very recent mixtures in the population, or could be because of founder events that are small populations long time ago. And indeed, in most of the populations, what we find is that it's because of small population sizes in India because of endogamous marriages. Now, one consequence of founder events or endogamous marriages is that because of random sampling of the population and a subdivision of the population, sometimes if you consider, sometimes you can have a particular variant increase in frequency just because of random genetic drift. Now here what I'm showing you is just a cartoon figure of this. Let's imagine at a particular locus, you have these red variants that increase your risk for disease and the yellow variants that do not increase your risk for the disease. Now, individuals might each be carrying one of these variants at a particular position. And then when they form a smaller population, what could happen is randomly there could be more individuals who carry the disease variant present in the subpopulation. And as the subpopulation expands, now this disease variant is present at a higher rate in the population. Indeed, we do see examples of this in our data set. One such variant is this variant that is present in butyrylcholine esterase gene. This variant in itself does not actually lead to any deleterious effect. However, when individuals who carry this variant get an anesthetic drug, which is widely used in India, individuals carrying this variant can have severe deleterious response, have muscle paralysis in response to this drug. That's because this drug cannot be metabolized if you have this mutation. Now, this mutation, which is so deleterious, is actually seen nowhere outside of South Asia. This is a plot from Meade, which has 150,000 whole genome sequences in the study. And if we look at just the frequency of this variant in the Meade data, we find this variant is not present in that data set. However, in South Asians, it's present at 0.3% frequency, again at very low frequency. However, in some groups in India that live in the south of India, this variant can be present at over 10% frequency. In one group called the Vaishya, this variant is present at a high frequency, and this is because of potentially the founder event that occurred in this group, that this deleterious variant sort of randomly increased in frequency in the population. This is not just a single variant of this form. There are many other variants that are deleterious that have increased in frequency because of founder events, some related to, say, cystic fibrosis or hearing loss, or some mutations in hemoglobin genes have also similarly carry very high deleterious effects, but because of founder events may have increased in frequency. Now, finally, I will tell you about what we are finding about Neanderthal and Denisovan ancestry in South Asians. So, like I mentioned, people living in Asia have both Neanderthal and Denisovan ancestry. So, if we look at Neanderthal and Denisovan ancestry in present-day Indians, what we find is that they have about 1.5% Neanderthal ancestry, and they have about 0.2% Denisovan ancestry. 0.2 is really tiny, but because our genome is so large, 3 gigabases long, we can, even 0.2% is actually several megabases long. What we then did is we tried to look at, did a fun exercise of trying to see how much of Neanderthal ancestry or Denisovan ancestry can we recover from our own genomes. Now, as I mentioned, that because of crossovers, different individuals have Neanderthal or Denisovan ancestry at different positions in their genome. So in this individual, if a crossover occurred here and here, they might carry Neanderthal ancestry here, but not in the next region. And then this individual carries, say, Neanderthal ancestry in this region. And so what we can do is, because we have sequenced so many individuals in these large biobanks and these large genomic surveys, we could just start combining different bits of Neanderthal ancestry across individuals. And when we do that, by doing that, we can then build a linear genome of Neanderthals just out of the little bits of DNA hidden in our genomes. And in doing so, we can recover a large fraction of the Neanderthal and Denisovan genomes from our genomes. So here on the bottom, I'm showing you Neanderthal ancestry, and then the top is Denisovan ancestry. As you may remember, Europeans have the same amount of Neanderthal ancestry as South Asians, about 1.5% per individual. So the largest survey of archaic ancestry or Neanderthal ancestry has been done in Icelanders, where they have sequenced 27,000 whole genome sequences. And using that data, you can recover about 1 gig of the Neanderthal genome, or 1 billion bases of the Neanderthal genome. However, using our Indian data which has a tenth of the sample size, we can recover almost 50% more Neanderthal genome from Indian genomes than from the European genomes. And this is because the Indian history is so complex that different individuals are quite different from each other and carry Neanderthal ancestry in different positions. So even though they have the same amounts of Neanderthal ancestry per individual as Europeans, they tend to harbor that in different regions such that we can actually recover more of the Neanderthal genome using their genomes. We can do the same exercise with individuals. We can do the same exercise with Denisovan ancestry, and we are able to recover about a quarter of the Denisovan genomes from living Indians. Europeans have very tiny amounts of Denisovan ancestry, and so in this case, we recover only about 0.1 billion bases, 0.1 billion bases of Denisovan ancestry from their genomes. What is really interesting is with Neanderthal ancestry, we are now recovering as much of the Neanderthal genomes from living individuals as we are from the direct sequencing of the Neanderthal genomes, because after filtering, we are only able to recover about 1.6 billion bases of the Neanderthal genome, which is the same as what we are recovering from present-day individuals. And this highlights that modern humans today are the largest repository of archaic ancestry. Now, finally, we turn to look at what is the function of these Neanderthal and Denisovan variants that we harbor in our genome. And to do this, we again looked, as I showed you in our ancient DNA samples, what is the frequency of Neanderthal ancestry, here shown in blue, and Denisovan ancestry shown on top in yellow. What is the frequency at every position in the genome, and each of the chromosomes is shown here chronologically. We can then look at regions where there is very high amounts of Neanderthal or Denisovan ancestry. When we do that for Neanderthal ancestry, we find a region on chromosome 3 where people living in India have nearly 50% Neanderthal ancestry. This region actually harbors variants that increase the risk of hospitalization post-COVID infection. This is because it carries many genes that play a role in immune function, including some genes that might be interacting with the ACE receptor of SARS-CoV-2. This variant is present at very low frequency in European individuals. We can also look at Denisovan ancestry, and in this case, what we find is that Denisovan, and there is a strong enrichment for Denisovan ancestry at the MHC, which is the major histocompatibility locus, which is related to many autoimmune diseases and infectious diseases, that harbors nearly 10% of Denisovan ancestry in South Asians, and likely because of certain variants that we inherited from Denisovans that allowed us to adapt to the diverse climates or pathogens outside of Africa. This region is also shared with East Asians, who also have some selection at this locus. So finally, just to summarize, I showed you that South Asians have both Neanderthal and Denisovan ancestry, and these have played a major role in shaping the immune function in present-day individuals. They also have both the Neolithic farmer ancestry as well as steppe pastoralist ancestry, and potentially, like in Europeans, the steppe ancestry may play a role in lactase persistence in South Asians. And more recently, there have been strong founder events that can sometimes increase the risk of disease burden in some groups because of just random drift. With that, I would like to thank my team and my collaborators. I've been extremely fortunate to have a wonderful set of graduate students and postdocs, as well as wonderful collaborators who make doing this science really fun. None of this research would have been possible without the support of all our funding sources, from the Burroughs Wellcome Fund to the National Institutes of Health. And thank you very much for listening, and I'm happy to take any questions.
Many thanks, Priya. An excellent talk, and I think all three stories were very interesting. I would like to begin with some of the questions we got regarding lactose intolerance. So the first question is lactose intolerance an evolutionary consequence of domestication, or is it a genetic flaw, and why?
Yeah, lactose intolerance or lactose persistence, it's really a very puzzling locus. So it is the strongest signal of selection in our genome. So if you look at the region surrounding the lactase variant, what you find is that there is a very clear signal of selection. There's a long haplotype. There is very limited diversity in the region to maintain this genetic variant, which is present at nearly 90% frequency in some groups like Europeans and some pastoralist groups. But what is puzzling is that we still do not know why this variant is under selection. It was hypothesized that this is because of dairying, and as humans started domesticating animals and having a source of dairy, this was a classic example of gene function coevolution, where this variant then increased in frequency. However, ancient DNA has unleashed some puzzles where we find that dairying begins about 7 to 8,000 BC ago, but the variant is not present until 4 to 5,000 years ago, and then it starts rapidly increasing. But by this time, the population is practicing Neolithic farming, and so there should be already a good source of food available. So another hypothesis is that maybe it's because of famines, that during periods of famine, milk was really critical in helping people providing a source of nutrition. Or it must have been vitamin D, which was providing that we got through milk in times of cold climates when there was limited sunshine. But neither of those theories probably fully explain things, because what we find is that for about 4,000 years after dairying, people are consuming milk, they are consuming cheese, they're consuming yogurt, and that's a fine way to use milk rather than just directly drinking milk. During famines, you often also don't have a source of milk because animals are also suffering equally. And what we found in our analysis in India really strikingly, and I didn't emphasize this much, is that we find that lactase persistence is likely only under selection in these two pastoralist groups we studied. In all other individuals in India, the proportion of lactase persistence is similar to the steppe ancestry. There's almost a nearly 0.9 concordance between the frequency of lactase persistence and steppe ancestry in individuals, suggesting that it's actually not under selection in South Asians, even though South Asians actually widely consume milk and milk products. So it's really puzzling what is the selective pressure to that allowed this variant to be at this frequency.
Yeah, that's quite interesting. So just for context, where Priya says that if the variant for lactase persistence was beneficial, then you would expect that variant to be at higher frequency than the frequency, or that the percentage contribution of the steppe people to the population, and it's very much the same. So I've heard that many of the Southern Europe cultures have used milk for a very long time, but that they do tend to process it.
Yeah.
So it's not the use of dairy per se.
Yeah.
It's the
yeah.
It's the practice of drinking milk.
Yeah.
That is completely different from being a pastoralist and consuming milk products or domesticating cattle.
Yeah.
Drinking milk seems to be something else that does not really come hand in hand with having cattle or using milk.
Yeah. But I think the puzzle is if you can just process the milk, why do you need to genetically adapt to drinking it? Like there are many ways of adapting and consuming and deriving nutrition out of milk rather than having to just drink the milk. So if you can make cheese, yogurt, which is widely seen across different parts of the world, there is no need for the genetic adaptation, which was also likely true in Europe, where for 4,000 years before this variant arose, they were consuming milk and milk products. Moreover, people who are lactose intolerant can still, like me actually, can still consume some amounts of dairy, but just not, large amounts of dairy. So a cup of milk is okay. A liter of milk probably will make me quite sick.
Yeah, well, that brings me to the next question. You mentioned that there is a different variant, a different genetic variant in African populations of pastoralist people. So, is this an independent variant? And does that tell us anything about domestication of cattle there?
Yeah, it is an independent variant. In fact, Middle Easterners also have another variant. So there are multiple variants that have been identified in different diverse populations. In most cases, it's been identified in pastoralist groups. So there is evidence that they were practicing dairying and domesticating cattle or other dairy producing animals. But all those mutations are actually present upstream of the lactase gene in a gene called MCM6, which acts as a promoter for lactase. And so the mechanism appears to be the same, but there are just different ways evolution has taken to achieve the same outcome.
Great. Okay. Well, it would be quite interesting to see how that variant has spread relative to the timing of domestication and the use of dairy products. So the next question is, why is it that there is a difference in timing for the arrival of Neolithic farmers to Europe and to India, and then the same for migrants of the steppes?
Yeah, that's a really interesting question. We know that Neolithic farming or farming is practiced in India since about 8 to 9,000 years ago. However, actually the gene flow occurs much more recently. It's an open question of why the gene flow occurs more recently. The timing of the gene flow is actually still quite uncertain. We tried to date that mixture of Neolithic farmers and hunter-gatherers in individuals that were actually sampled outside of India in Central Asia, but they had indigenous Indian ancestry. And so it looked like these might have been migrants from South Asia that were in Central Asia. And based on that, we inferred this date of 5,700 to 3,400 BC, but it's still quite uncertain. But it does appear that farmers and hunter-gatherers may have been living together for a long period of time before they started mixing. Why that is still an open question. And I'm hopeful we'll have more samples from South Asia which are deeper in time scale so we can really study the dynamics of this mixture. With steppe gene flow, the story is slightly different. Actually, what happens is the steppe pastoralists mix with Europeans about 3,500 BC ago. But then there is backflow to the steppe where there are groups living there that have European farmer ancestry. And it's these intermediate groups that then go to South Asia. So it's almost as if they went not directly from the steppes to India, but they went from steppes to Europe to back to the Russian steppes and then to India. And now there is so much sampling in this region that we can actually start studying these dynamics and really mapping the population movements.
Yeah, I think that, as you say, highlights perfectly why it is so important to collect as much genome data as we can from as many individuals as we can from a variety of places. I think along this series of talks that we had over the last few weeks, it has been highlighted several times, this oversampling of genome data from individuals with European ancestry. And I think Priya's research really shows how much we can learn if we start sequencing genomes from individuals in large quantities from other places. So we have other questions relating to this idea of positive selection. So from a genetic point of view, what is the evidence that humans are still evolving?
I think from a genetic point of view, you can look at it in many ways. If you look at what selection and adaptation really means is that a variant that you have increases in frequency maybe faster than it would by neutral drift. If it was just changing randomly every generation, you either have a chance to get it or not have it, and there is no effect, then that's what you might think from a neutral variant. But if it has a functional effect or if it impacts the fitness of a population, then it might increase in frequency a bit more rapidly. And we can see in these large biobank style data that there is some evidence of certain variants that are increasing in frequency. There is while we study selection and adaptation a lot, what we find is that on a genomic scale, there are a handful of variants where we have really been able to identify signatures of selection. And so what we find is that as our environments are changing, as the pathogens are changing in our environment, humans are still adapting to these different environments. Just the changes and shifts are not potentially as dramatic as you see with, say, lactase persistence.
Yeah, thank you. I think that's a very good point. Strong evidence for positive selection is actually quite rare.
Yeah.
I do comparative genomics on other species, and it's very hard to pinpoint those areas. So going to your first story, do you find any Neanderthal gene deserts in the human X chromosome?
Yeah, X chromosome is really interesting. In fact, I will share this slide. X chromosome is actually really interesting because it has very large gene deserts. This had been identified already in present-day individuals, but when we looked at our data with ancient DNA samples, here again are sorted by age, DNA samples at the top, present-day samples across the world, and what you find is actually there are very large deserts of Neanderthal ancestry across the X chromosome. And in fact, there are only the X chromosome that can tolerate Neanderthal ancestry. And some of these regions also they overlap some of the selective sweeps in non-African. And it's really an open question of why there are such large deserts, why Neanderthal.
Do you have any data on the Neanderthal genome, and are there any similarities in the patterns?
Yeah, I think this is a very active area of investigation. It's a more challenging question because we only have 3 high coverage Neanderthal genomes. And so it's been harder to pinpoint if there was gene flow from modern humans to Neanderthals, though with more sophisticated methods, it's becoming clear that there may have been gene flow from modern humans into Neanderthals also. Although with 3 samples, it's been harder to sort of look at the frequency distribution and the patterns in as much detail.
Yeah, thank you. So we also have a question about the genes related to language. I think this question comes from one of your very first slides. So it says, which genes are related to language ability in humans and how did they evolve?
Yeah, so one of the most interesting genes related language ability is FOXP2. This is a gene on chromosome 7 where there are 2 non-synonymous variants that are present that were identified first in individuals who had language that had issues with language development. And so it was associated to language development. When the Neanderthal genome was sequenced, they identified that actually Neanderthals also have these same non-synonymous variants. They are not seen in chimpanzees, but they are actually present both in Neanderthals and Denisovans. And this might suggest that they too had similar language ability as modern humans. What is really also interesting is that one of the deserts, Neanderthal ancestry deserts that I showed you, is on chromosome 7, and that actually includes FOXP2. It's a 10 megabase region. It includes many different genes, but one of the genes it also includes is FOXP2. And so it still remains unclear what are all the genes related to language, given that I would think it's a complex trait like height or skin pigmentation, many variants might be associated to it. But one of the ones that we know is FOXP2 from sort of surveys of present day as well as present day samples and also some functional work.
Okay. So we also got a couple of questions on whether we can bring back extinct species. I think that these questions are coming very frequently on this series because of the recent news of bringing back the wolf terrible.
Yeah.
So it says, can the DNA be a tool to study why species get extinct? And and the second one is, can genetics bring back an extinct species?
Yeah, I mean, I think I'm probably not the right person to answer. Can we bring back different species, since I mainly focus on computational analysis, and I would think this is more of the genome editing, experimental analysis side of things. I'm not sure if we should be bringing back species. I often teach undergraduates human genetics every year, and I show them sort of, what might be a way that you can bring back different species. How might you clone a mammoth, for instance, the famous book by Beth Shapiro. But then I often end that discussion by showing them a photo of Jurassic Park where, maybe this was not a good idea to have brought back those species. And so I think I will leave the experts to decide if and how we bring back species. What I do think genome data can do is help us understand why some species went extinct and why others are here. And in fact, this was initially the motivation for ancient DNA studies in Alan Wilson's lab, where he was really interested in understanding why the quagga species was extinct. And so I think this can help us understand why some species went extinct. It can help us also understand what our past was like. Who were our ancestors? What did they look like? What were certain phenotypes that we inherit from them? But I'm not sure if we really want to try bringing them back.
Thank you. So we are running out of time. Thank you very much. And we're going to finish with one last question relating to your last story. And that is whether you found any correlation between genetic similarity and language similarity in the subcontinent, or whether geographic distance plays a more important role.
Yeah, I think the Indian subcontinent in this case is very interesting because different groups in India live. There is sort of a population structure in across India that is geography, as well as the find that there is higher step ancestry north of India. That is also where they speak more Indo-European language. South of India, and similarly, there is stratification along those dimensions, but they are quite correlated. And given that there is now endogamous practices, sort of patterns that may have been wiped out over time are in some ways frozen through, and so all this complex structure. And I think this is one of the reasons why we see so much diversity in Indians also.
A lot of population structure. Thank you very much. I think we all enjoyed it very much. And we thank you for your time and the very interesting conversation we had afterwards. And all that is left to me is to pass the stuff to Dr. Jaime Urrutia to close the session.
Thank you very much for inviting me. Muchas gracias.
Yeah, I think. Oops.
You are on, Priya. Can you turn it off? Maybe that's why we have. Thank you.
Okay, yeah, thanks very much. I think we solved it. Well, thanks very much, Priya. It's a really interesting presentation, and congratulations for your studies. It's amazing the resolution you have, and adding, as Araxi mentioned, the studies in other parts of India, other parts adds covers the complexity in evolution. We are very grateful for the presentation, and it's amazing the resolution of the studies. I think having this ability of dating the evolution, what has been happening through the years, is really very, we are very, now we are very curious to learn more about your research. Thanks to Araxi and Majal for organizing this seminar cycle, and thanks to all of the you that have been joining us and for the comments and the questions. And we thank the offices of the university in the US, Canada, UK for joining us and retransmitting the seminar cycle. And thanks to the students, including the high school students of the university who have been joining us for the different presentations. And we invite you to the following conference by Machal, and we look forward to invite Priya again and learn more about what the new studies she's doing with her group tell us about the human evolution. Muchas gracias a todos por acompañarnos a lo largo de esta serie. Ha sido una aventura fascinante el ver la resolución que se tiene para trazar nuestra historia. Cómo podemos ver con ese detalle cómo ha sido la evolución y el resultado de las migraciones. O sea, toda la población humana ha sido construida a lo largo de los grupos que han migrado a las diferentes partes. Cómo esto se puede registrar con ese detalle en el genoma. El genoma es realmente nuestro mejor libro para entender cómo es la evolución de los diferentes grupos a lo largo del tiempo. Y si algo nos puede esto decir, pues es la importancia que ha tenido la migración en construir la historia de la humanidad, en construir lo que ahora tenemos como las diferentes culturas y lo que tenemos ahora como sociedad humana. Y lo que nos enseña también es que a lo largo del tiempo estas culturas se han mezclado en diferentes formas. Somos el resultado de una mezcla que ha enriquecido a todos los grupos humanos. Thanks very much, Priya, for telling us how you can uncover the history of the human cultures. And it's fascinating to learn how it has been built from the groups that have migrated through the world. And we are a society built on these groups, on these migrating groups. So it's really interesting to learn more about this. And you have left us with a lot of questions, so we will be very much interested in having you back in this seminar cycle. Thanks. Muchas gracias y pasamos la palabra a Dionisio. Muchas gracias a Araxi y a Machal por esta serie tan fascinante de conferencias.
Muchas gracias, Jaime. Muchas gracias, Priya. Decía Jaime que nos dejas más preguntas. I think that's a good balance. These sessions. Tenemos más preguntas que respuestas al final. Quiere decir que fue una exitosa. Thank you very much again. Esperamos seguir contando con ustedes de trazo evolutivo que necesita conocer más de la propia historia. Eso significa un avance importante para conocer cuál es el rastro de la humanidad, el rastro de su presencia en esta tierra, dado que debemos de tocar esta especie, las demandas, ese tanto. Gracias a toda nuestra audiencia. Vamos a seguir contando con ustedes la experiencia fascinante, evolutivo, haciendo gracias a la de Araxi, de Urrutia, y ahora con la. Muchas gracias a todos y
Hemos becado a más de 1 millón de estudiantes universitarios.
Más de 84,000 alumnos y alumnas han sido beneficiados con becas nutricionales.
Lanzamos una campaña para apoyar a la Universidad Autónoma de Guerrero, afectada por el huracán Otis.
Este año organizamos nuestra tercera gala musical y nuestra cuarta carrera atlética.
Estamos felices porque más del 87% de nuestros becarios son la primera generación de universitarios en su familia.
Cada año se apoyan a 100 estudiantes de la Facultad de Derecho.
Hemos entregado más de 600 becas a estudiantes de bachillerato junto con Fundación BBVA.
Hemos transmitido 195 documentales y 160 conferencias.
Apoyamos a la Unidad de Investigación en Neurodesarrollo de la UNAM.
Todos nuestros informes son públicos.
Se han entregado más de 50 premios para fomentar la investigación científica.
Con el programa Bécalos, el Patronato de la Facultad de Química y Fundación UNAM otorgaron más de 30 becas.
Con la ayuda de Fundación Carlos Slim hemos otorgado becas de apoyo nutricional y económicas.
Con el programa Hídrico Modelo ayudamos a 340,000 habitantes.
Tenemos 2 premios EFI Social por nuestras campañas publicitarias.
Junto a Grupo VAL, más de 2.000 universitarias han sido beneficiarias.
Otorgamos más de 400,000 tratamientos.
Durante la pandemia apoyamos con las campañas Dona un Kit y Apoya a un Residente a estudiantes de medicina de la UNAM.
Durante el confinamiento COVID, donamos tablets para estudiantes de escasos recursos.
Hoy son más de 27,000 solidarios impulsando la educación.
Participamos en la conservación de corales en el Caribe mexicano.
Brindamos más de 4,000 becas de movilidad internacional.
Colaboramos con el Semillatón a preservar el maíz en la Sierra Tarahumara.
Apoyamos el proyecto de conservación de perritos llaneros en la Reserva de la Biosfera de Jano, Chihuahua.
Participamos en el proyecto de conservación de la guacamaya roja en el Parque Nacional de Palenque.
Desarrollamos el programa Líderes Transformadores.
Participamos con el Consorcio de Universidades por la Ciencia.
Organizamos 11 ediciones del Foro 2020.
Fundación UNAM resguarda el Palacio de la Autonomía, inmueble histórico de la universidad.
A través del CI ofrecemos cursos, diplomados y talleres.
Dotamos de equipo al Observatorio Astronómico Nacional de San Pedro Mártir.
Apoyamos a alumnos de escasos recursos.